All right. Hello, guys. This is the beginning of the Module 6 online lecture covering bacterial transformation. As most of you should know, this is asynchronous, so you can watch the video lecture, which this is, at your leisure. Um, just a little disclaimer, I've already attempted recording this like 6 or 7 times and I'm not going to be editing it. So if something weird happens or I can't figure out how to go to the next slide, just gonna have to bear with me. So we will begin by talking a bit about what is going to be expected of you for Module 6, and then I'll lay out kind of what you need to prepare for the next and final lab. So first being pre-lab 6, you should have this completed in your laboratory notebooks. You don't have to email me images of this. You'll be turning in your lab notebooks next week. So I can check to see if it's completed at that point. For Module 6, while you won't be physically completing the procedure, you're still going to want to download this and detail the methods in your laboratory notebook. So this kind of coincides with the last point here. And as we go through the slideshow, I'll be providing a lot of the background information and detailing what you would have done if it was in person. So what you'll include in your notebook entry and some answers to the discussion questions will become a little bit, ideally, more clear. You will still be doing worksheet 6 as well. This won't be due until the next in-person lab, so you don't have to worry about turning this in next week. Then quiz 5, which would be on the second part of enzyme kinetics, will be posted to EDUCAT on Friday, and you'll have a week to complete that as well. Make sure you do review the material. I'll do a bit of a quiz review, but it is going to be timed, and I don't want anyone to bomb it. So for next week, you're going to turn in your laboratory notebook. So fine tune your modules. If you haven't included your results or haven't answered all the discussion questions, or maybe you need to add a little bit more in your background or methods, go ahead and do that for all of the previous modules. If you forgot what you should be including, there's been a notebook requirement sheet posted to EDUCAT all semester, and you can feel free to look back at that. If you have any questions, I do have office hours Tuesday from 11 to 1. If that time doesn't work for you, feel free to reach out. We can meet a different time. And if an influx of people seem to have questions, I can hold maybe an open lab or some more Zoom office hours. The next week, or maybe 2 weeks if you're in subsection B, we'll be doing a central dogma assignment. So this will include kind of a take-home essay exam. It wouldn't be timed, of 5 questions, and you'll be able to do them at your leisure. And this will be due at the end of the semester. And then in class, we'll be doing a bit of an in-class assignment related to gene bank databases, and that you will need your computer for. So make sure you bring that. I think that's all I have to say about that. So for the quiz review, let's get right into that. The first question I would have for you is, what was manipulated in the last enzyme assay? So if you remember back to the very first week of enzyme kinetics, we manipulated enzyme concentration with both our purified and our isolated acid phosphatase. So in the second part of the enzyme kinetics, we manipulated substrate concentration. And what did we calculate from this data? We calculated 2 values, and that was the maximum velocity and the Michaels constant. So you should have had a bit of a graph that looked like this, and this was kind of an intermediate step in our data analysis, where this value right here where we're seeing the plateau would be our maximum velocity. And within the linear region of that, when we were to calculate or approximate just from eyeballing the graph, half the reaction rate at half of the maximum velocity, that substrate concentration would be our Km value. We'll talk a little bit more about those in the next slide. So what does the Michaelis constant tell us, the Km value? This is an index affinity of the enzyme that the enzyme has on the substrate. This is numerically equal to the substrate concentration at which the reaction rate is half the maximum velocity. So if you think back to last week and the concentration of our substrate, that was in micromolar. So your Km value should be reported in a micromolar with a micromolar unit. So what does our Vmax tell us? This tells us the maximum velocity of the enzyme at which the enzyme is fully saturated with the substrate. So if you remember at the Km value substrate concentration, the enzyme, half of the enzyme is in complex with substrate, while at the maximum velocity, the enzyme is saturated by substrate. So all of the active sites are bound to substrate. So I already talked a bit about this. What are the units for both of these values? The Km value is going to be reported in micromolar, while the Vmax is a velocity, which is nanomoles over minute. And that nanomoles is the p-nitrophenol, which is our product. Okay, and then just a quick review of the Lineweaver-Burke plot. In order to accurately determine the Km and Vmax value, we set that y-intercept, your graph here, it's labeled C, the y-intercept here is equal to 1 over the Vmax. So we were able to solve for the Vmax, and then the Km over the Vmax is equal to the M value, your slope. So once you determine your Vmax value, you can plug it in here and then solve for your Michaelis constant. Okay, so to get started on the lecture material for Module 6, we're going to talk about some horizontal gene transfer. So horizontal gene transfer is the ability bacteria have to accept foreign DNA or DNA that is not their own during their life cycle. So we have 3 different forms of this. We have transformation, which is what we would have actually been doing in class. And this is the process that bacteria will take up foreign genetic material. So this is in the form of plasmids. We have transduction, which is the process by which foreign DNA is introduced to a cell by a virus or a viral vector. So that's here in the second image. And then we have conjugation, which is the ability for bacteria to transfer DNA. Again, this is going to be in the form of a plasmid through a pilus. So this is a direct contact from one bacterium to another. So for bacterial transformation, again, this is a horizontal gene transfer. So this is going to be genetic modification of the bacterium through the uptake of extracellular genetic material in the form of plasmids from the surrounding environment. This is going to be through the cell membrane. In order for this to happen, cells must be competent, which means the cell membrane is permeable to the DNA. Cell competence. So again, in reference here to the cell must be competent. This confused me when I was first learning this material because I thought it just meant that the cells were living, but it means that they have a permeable membrane here. So this can be natural or induced competence. In natural cases, going to be in direct response to environmental conditions such as cell starvation or cell density. Induced, so in our case, we would have induced cell competence in order for it to take up the plasmids that we were introducing to it. So this is going to be through the treatment of calcium chloride and a heat shock. So what are plasmids? They're small double-stranded circular extrachromosomal DNA. So kind of here in this image, we're seeing bacterial DNA, but then we're also seeing these significantly, this is not drawn to scale, plasmids are significantly smaller, and they're circular, and again, they're extrachromosomal. Naturally, plasmids only contain one or a few genes that are going to be useful for the survival of a specific environment. So if you can kind of think of some environments that bacteria might be facing that would be dangerous to them, we could think that genes of antibiotic resistance might be useful, or maybe the ability to metabolize certain nutrients in their environment in cases of high cell density. So if you're a bacteria, and say you don't have the ability to digest lactose and use it as energy, but then you are in a situation where you're in a extremely high cell density, then the ability for you to uptake a plasmid that allows you to digest lactose may be very advantageous in the case that nutrients are sparse. Artificial plasmids, though, are frequently used as vectors for molecular cloning, and they allow the replication of a specific recombinant gene. So we've really exploited bacteria's ability to uptake plasmids and start coding for whatever gene is on the plasmid. So a lot of chemicals and medicines and what have you are manufactured in this way. Some examples of this would be insulin. Insulin pretty much exclusively is grown up in non-pathogenic forms of E. coli and other bacteria. Casein, which is a protein used to make cheese that comes originally from the intestines of cattle, I believe, is also grown up in non-pathogenic bacterial cultures. And when we transform a bacteria with a plasmid and it starts coding for our protein that we have on the plasmid, we're able to then purify the culture, and we're able to purify the specific protein of interest. So say you transformed a E. coli cell with a plasmid that contained an insulin gene on it, then that bacteria, we're going to be exploiting its cell machinery. It's going to start coding for that insulin. It's going to make insulin protein hormone, and then we'll be able to isolate that insulin from the cell culture altogether. And then we have our product. Prior to growing it up in a cell culture, we would isolate it from animal pancreases, which is incredibly expensive to do. If you think you have to grow up like a pig, for example, that was where most of our insulin came from, and then euthanize the pig, harvest its pancreas, and then extract the insulin from that. This is a much quicker and cheaper way of doing it. Figure out a way to edit this. Okay, so if we would have been doing this lab in person, you would have been working with 3 unknown plasmid samples. They would be labeled A, B, and C, and each one would contain a different plasmid sample. So one plasmid sample would be PUC19 wild type plasmid. One would be PUC19 recombinant plasmid, and one would be just water as a control. So we would transform cells, E. coli cells, with this plasmid, and then we would do what is called blue-white screening. From there, we would run an enzyme digestion of the plasmids, and we would run them through gel electrophoresis. And based off of both the blue-white screening and the plasmid digestion with the gel electrophoresis, we will have been able to effectively determine what plasmid we were working with. So, pause here. Okay, so the PUC19 plasmid here is 2,686 base pairs long, and this is going to be what's referred to as the plasmid backbone. And it has three, I guess mainly two really important genes in it already. So we have the LAC-Z gene here, which will code for an enzyme called beta-galactosidase. This enzyme gives bacteria the ability to digest lactose to galactose and glucose. I'll talk a bit more about this in the next slide, but it's also going to have an ampicillin resistance gene. And this is going to be very important for knowing what cells were successfully transformed. I'm going to talk a bit more about the beta-galactosidase, and then we will come back to this. So beta-galactosidase will also cleave a molecule called xGal, which is chemically similar to lactose. When there's cleavage of the xGal molecule, it will turn blue. The colonies the colonies of bacteria will physically turn blue. So xGal, along with ampicillin, are compounds that are going to be added to the agar that we're growing our bacteria on. So effectively, only the cells that were successfully transformed with the PUC19 plasmid will be able to grow at all, because if they were not transformed, they won't have this ampicillin resistance gene, and the E. coli will be killed by the antibiotic. Now, only the cells with an intact LAC-Z gene will produce that blue pigment. So this will allow for blue-white screen. Now, you may be wondering, if we're just here working with the PUC19 plasmid, shouldn't all the bacteria be turning blue? And the answer to that question would be yes. The wild-type plasmid, which this is right here. Let me make sure that you can see my cursor, which is right here. All of the cells that are transformed with the wild type PUC19 plasmid should have should turn blue, because they have an intact LAC-Z gene. So they'll have the beta-galactosidase enzyme, and they'll be able to cleave xGal. Now, this polylinker right here, I have another slide on it, will allow for a recombination of another plasmid. So you can see here at the EcoR1 sites in between both of them, we're seeing a thing that is called the multiple clone site. So here we're able to recombinate a gene into the middle of the LAC-Z gene. Now, when we do that, we will be disrupting this LAC-Z gene, so beta-galactosidase will not be produced. Therefore, we will see a lack of blue colonies. So here in the bottom image, we're seeing the plasmid backbone, and then we're seeing the recombinant gene insert, and then we're seeing the EcoR1 cut sites on either side of that inserted gene. So the cut site is still in existence when we don't have a recombinant gene added to the plasmid. So this will be relevant when we talk about plasmid digestions, which will be coming up. Okay, so here we're seeing some blue-white screening. So we're seeing blue and white colonies. So that would mean that we are likely transforming a recombinant plasmid solution. So not every single plasmid in our solution would have been successfully recombinated. So we know which plasmid was successfully recombinated, in which case was successfully transformed by the color of the colonies. So the blue colony here did not, that plasmid that was transformed into that bacteria did not have the recombinant gene. Okay, let's go to a casein protein situation. So let's say we're growing up the protein casein in E. coli culture. We need to create a recombinant plasmid with a casein gene. So when we do that, and then we transform our bacteria. So the white bacteria here would have the recombinant plasmid and they would be producing casein, while the blue colonies here, they were transformed, but they were transformed with a wild type version of the plasmid, not the recombinant version of the plasmid. So B here, we're only seeing blue colonies, and that means we are working with the wild type plasmid. Now C, we're seeing no colonies, which means no bacteria were transformed. And in our little lab setup, this would be the plasmid solution we were working with was just water. Okay, so now moving on to plasmid digestion. So plasmid digestion utilizes restriction enzymes to cleave the DNA at specific sites called cut sites. Our restriction enzymes cut at the EcoR1 site, which is at the 396 base pair mark. This will allow us to be able to identify our plasmids, the wild type and the recombinant. Obviously, on the water, nothing will show on the gel. So, like I said before, in the case that we have a recombinant plasmid, we're going to have two cut sites here. So when we cut at both sites, we're going to see two bands appear on the gel. Now, in the case of the wild type plasmid, we're only going to have one cut. So you're just going to see one band on the gel. So here is an example from EDUCAT. This was a gel from last semester. So here in lane 2 and in lane 5, we're seeing two bands. So this would be an example of a recombinant plasmid. Here, the downstream band would be the recombinant gene, while up here would be the PUC19 wild type plasmid backbone. Now in lane 4 here, we're seeing just the wild type plasmid, which is appearing about in the same position as the upstream bands in both lane 2 and 5. So if we're thinking back to here, this lane would have to be plasmid sample A, again, the recombinant sample, the same one we're getting blue-white screening. Lane 4 here would have to be B, just the wild type version of the plasmid where we're just getting that blue color appear. And then lane 3 here would be C, and that would just be water. So that concludes the lecture I have for this portion of the class. Now I'm going to go onto the EDUCAT page, and I'm going to show you some of the resources that are available there. So here we have Module 6 posted and the pre-lab. Again, download these, read through them. Same with the worksheet, and complete them before your next in-person lab period. Then we have a series of 4 plasmid digest gels. You can pick any one of them and include it in your notebook and use it for your analysis. Here is the one KB ladder that you're going to need to include with these gels, so you know what that standard lab ladder is representing. Then here is a file for some supplemental materials. So here is some greater explanation of the restriction digest, and there will also be some images for blue-white screening that you can include in your notebook as well. Then these last 2 are videos. So this is going into a little bit more detail about the mechanism of transformation for competent cells. And then this last one is blue-white screening for DNA isolation. Yeah, so like I said, you can pick any one of these gels for your data analysis. Okay. All right, so that concludes what I have for this lab. Feel free to email me if you have any questions specifically about data analysis. I'm sorry that this lab had to have been online. I know some other instructors were doing in-person Zoom, well, not in person, but synchronous Zoom. So you met during your lab time. Because there is a Wednesday section, I didn't, and they weren't able to meet for the Wellness Wednesday. I decided to do all of them asynchronous. If you do have questions, feel free to reach out. Again, I have office hours Tuesday from 11 to 1, and I can always schedule a Zoom meeting, or you can swing by, just email me so I can make